honor and pleasure to introduce Mitesh Borad today to the Liver Cancer Program Seminar Series. Mitesh received his BS at Biomedical Engineering from Boston University and then an MD from Rutgers, New Jersey Medical School in 2000. He completed his training in internal medicine at Cedars-Sinai and medical oncology at Tulane in New Orleans. He was a genomic medicine and drug development scholar at the Translational Genomes Research Institute in Phoenix, and then trained with Dan Van Hoff. His research focus has been and continues to be the development of novel therapeutics in liver, biliary, and pancreatic cancers, leveraging both precision medicine as well as gene and virus therapies. His group was the first to introduce an oncolytic rhabdovirus into human clinical studies, and further development of this exciting area has been the focus of this efforts, although he says that today he will be talking something else. So without further ado, Mitesh, I'm very happy to listen to your talk.
Okay. Thank you, Tim. And it's a very distinct honor to be here with this August group. As I mentioned briefly, I have done a fair amount of work in oncolytic viruses, but the talk today will be primarily on precision medicine. Okay. So hopefully everyone can see my slides. So I'll talk a lot about the bedside-to-bench interface, and what this entails is doing initial discovery at the bedside. And I don't need to convince many in this group. I just had a chat with Tim earlier today, and he says he is also a big fan of this approach. So hopefully I can highlight, some of the advantages of this as we go along. So here are my disclosures that come from working with many biopharma companies. So the objectives will be to use biliary cancers as the model here and gain deeper insights into known targets for biliary tract cancers, demonstrate how these kind of bedside-bench, back-to-bedside platforms can simultaneously allow for discovery and application as it pertains to precision medicine, but this approach can be applied to any arena, including immunotherapy. We know that there is heterogeneity of response, and we shouldn't be surprised as we dive deeper into these genomes and ways in which we could potentially tackle this. I'll touch briefly on liquid biopsy as a tool that people are already using, but it's pretty clear how it will be used in the future, maybe some discussion on that. And finally, I'll talk about the battle royale between evidence-based medicine and N=1 medicine. Okay, so biliary cancers, second most common primary cancers of the liver. Primarily, these are adenocarcinomas, although you could have some squamous or mixed tumors as well from a histology perspective. These are also highly aggressive and lethal, caught late in the game in most patients, where you can't use resection or transplant as treatment modalities and have to revert to systemic therapies. Even with all the efforts we have ongoing, unfortunately, as a group, the median survival is still hovering around a year. Many risk factors have been identified, including infections such as Opisthorhis viverrini or Clonorchis sinensis, which are the liver flukes in Southeast Asia, hepatitis viruses, salmonella for, say, gallbladder cancers, and then autoimmune conditions such as NASH, NAFLD, and PSC, especially in younger patients. There has been some observation that the incidence has been increasing both overall and particularly of intrahepatic cholangiocarcinoma. It seems like a constellation of better identification. A lot of unknown primaries are being correctly classified now, and there seems to be some real increase due to the same risk factors that have led to other obesity and metabolic disease-related cancers increasing in incidence. This is not a common disease in the Western world. You can see 1 to 2 per 100,000 incidents, shown in blue there. In Asia, excluding the hotbed area of Southeast Asia, it's 6 to 8 per 100,000. This is primarily due to the hepatitis viruses. And in Southeast Asia, because of a carcinogen, which are the liver flukes, it's 85 per 100,000. We should not view this as one disease. These are obviously distinct diseases just from the etiology perspective. And although they have a common terminal finding at the histology level, we are talking about a grouping of disease under the same term. Okay, so precision medicine is, of course, highly talked about today. But like anything else, when there's initial attempts at it, there's always skepticism. The search for a magic bullet has been ongoing for many decades. Shown here is Paul Ehrlich's search. This was not for cancer. This was for infectious diseases like syphilis. So we come to the concept of just rethinking how we look at cancer. So the microscope has been the primary tool for cancers. You can see the cholangiocarcinoma cells there, and there's of course many other supporting cells there and the immune cells. So really the tumor is an environment, but all of these ultimately would just be called cholangiocarcinoma, and the level of detail that you get with decision making is extremely poor if you just use this approach. So the new era obviously involves the genomoscope, and shown here is a whole genome representation of a tumor. There has been some work done with whole genome analytics. In fact, what we think is the complete map of the whole genome was only recently completed after filling in for some of those areas that had not been sequenced sufficiently. So we moved into an era of tumor classification where we started doing a lot of exome sequencing, and in the clinic, a lot of panel sequencing, and those were just retermed comprehensive genomic profiling, but they're far from comprehensive. They're only representative of 0.1 to 0.5% of the genome. So this is maybe a closer representation with mutations, rearrangements, and copy numbers, all shown on what we call a U-plot. So chromosomes 1 through 12, and on the right side, 13 through 22, and X and Y shown there as well, all shown on one plot, a little bit different than Circos plots, where you can also represent whole genomes, but they're hard to navigate for users. It hasn't been that long from when clinical genome sequencing has been in vogue, about 13 to 15 years. Holding that Circos plot is Jeff Trent. This was the first solid tumor clinical genome that was sequenced in a collaboration between Mayo and TGen in 2009. So not that long ago that this was considered very experimental. So when we talk about molecular profiling, people ask, what patient should you do it in? And the answer is every patient. There's no reason you shouldn't do molecular profiling, and one shouldn't really think about sort of reimbursement or utility and these types of aspects, because you never know when you're going to need that data. And it is now affordable enough, at least in certain geographies, that instead of trying to pick and choose which patients you should do this in, you should do it in everybody. So one way I found to be helpful to remember is just designated as the 6th vital sign. So you do blood pressure, pulse, temperature, respiration. If you count pain as the 5th vital sign, you add this as the 6th one, you'll never forget to do it. When you talk about profiling, as I said, you can look at many different aspects. You can just look at a few alterations through panels. You can do whole exome. You could do whole genome. You could extend it to whole transcriptome and other omic modalities, or you could do a more limited evaluation with array CGH or FISH, or if you want to do some preliminary protein analysis, immunohistochemistry. These are some of the early publications using exome sequencing to identify alterations in the disease. And you can see that BAP1, ARID1A, and PBRM1 were identified in one of these publications from the Western world, and a number of other alterations like P53, SMAD4, and KRAS were identified in the liver fluke associated disease. But these were small numbers of patients, and these were still just exomes. So you're talking only about 1 to 2% of the genome and not the full array of alterations that can occur. So bedside to bench, the patient is your laboratory, and if you have extremely robust, high resolution tools, you can start with the patients, see what you find, and then if you are going to do further investigation, you're not wasting your time anymore because your hypothesis is actually founded in reality. So as I mentioned, you have the omics tools, but what you're looking at only gives you a partial representation, and the elephant and the blind men example illustrates this very clearly. So what is the correct representation? Well, using something that uses many or all the tools will give you the full idea of the elephant. When you do more comprehensive sequencing, there are many advantages. So right now, people do separate analysis for germline, separate analysis for pharmacogenomics, separate analysis for therapy selection, perhaps a separate analysis for response monitoring, et cetera. This can all be coupled into the same assay if planned correctly. And obviously that would entail modalities like whole genome sequencing. Now with the prices coming down considerably, the challenge is not so much in the sequencing cost, but in the ability to analyze these if you have robust tools. Okay, the question that often comes up is when you look at a lot of data, how do you know if you have a driver in your hands? So there's a few sort of rules one can ascribe by. So one rule I like to use is multi-platform concordance. If you have a few different omics platforms and they all point to something being a driver, then there's a higher likelihood. If you look at a lot of cases and you see recurrence of the same event at the exact level or the gene level or the pathway level, again, that might be a driver. If you look at the transcriptome data, if there's allele specific expression of the mutation, that could give you some clues. You can rank certain kinds of events. So copy number events, you can rank from high to low. You can't really rank mutational events that much, but copy number events, you could use ranking. The highest events might be the highest likelihood of being drivers. And then sometimes a target is selected by having a mutation and an amplification, or a mutation and a deletion. And these, in my mind, also point to drivers. So many other people agree with these approaches. Shown here is an example of a patient from one of those early studies that I described with my colleagues at TGen, where we have data from a patient who had exome sequencing and RNA sequencing done. This is before FGFR2 fusions had been described. So we saw evidence of a fusion by both exome and RNA-seq. And although this was one of the first patients seen with an FGFR2 fusion, we were confident that this might be a driver event based on platform concordance. So we went ahead and gave this patient ponatinib. There were no FGFR-specific inhibitors available then. And you can see that there was some tumor necrosis and disappearance of lesions. And this happened in 2 or 3 other patients who had fusions. And as such, with just a few patients, we were convinced that these are driver events and may represent a cohort within this disease. So several other groups, both in the US and Japan, describe these events. So as I said, it doesn't take that many events to dive in deeper. So the target was discovered. Now what was needed? Maybe cheaper diagnostics, models where you can test out combinations and other hypotheses, both at the in vivo level and in vitro level, proof of concept clinical trials, and understanding target biology. This was fibroblast growth factor receptor 2. A lot is obviously known about it from a physiological standpoint. When it's altered, what happens? The disease is what can inform things in this case. So here's the pathway, receptor ligand interactions being key. So you have the fibroblast growth factors, which would bind to the receptor. This would lead to dimerization, activation of the scaffold with mediators like FRS2. And then you would have activation of the growth, proliferation, angiogenesis, cell survival pathways such as RAS, RAF, MEK, ERK, and PI3, AKT, and others. What is the prevalence of this event? We now know it's 10 to 15% in patients with intrahepatic cholangiocarcinoma, seen both in Asian and Western series. Cheaper tests could be something like a FISH. So given that it's a fusion, you can develop things like a break-apart assay. So this is a patient with a fusion where the probes, instead of aligning and forming a yellow, have distinct red and green. And as you can see here, just a few years after the initial description, we were able to launch a CLIA test in 2015 to do this. So now you can do this off of very small samples even, and you can deploy it to countries where sequencing is yet not common. Mark Trudy and Lewis Roberts, shown here, have had a long-standing effort of using the surgical suite to collect tumor samples from resections and generate patient-derived xenograft models for precision medicine. These can be, of course, quite valuable as tools that recapitulate many features of human disease. And what had not been done at that time was characterization of these tumors. So we characterized a few of these tumors with RNA-seq or ACGH, whole exome, and other modalities. And one of these models was shown to have an FGFR2 CCDC6 fusion. So now we have a preclinical model system. So you can see it has the same features on the break apart as shown here. The tumor can be implanted here subcutaneously. Of course, models where you orthotopically implant them in the liver can also be developed in future generations. And when you test drugs in these models, you can see that they have effect just like we do in patients, where here's vehicle treated, and you can see ponatinib, which I gave to that patient, had effect. And actually this tumor remained sensitive even to standard drugs, so provides a good context. When you use some of the FGFR specific inhibitors, you can see that they are more effective than ponatinib, which is what we had used in that patient. And of course, this is just one model, but as I'll show you later, these findings were shown to be relevant in a larger group of patients in proof of concept studies. In terms of rapid diagnostics, AI, machine learning, and other modalities are coming into play. And my colleagues from radiology and AI and machine learning assisted with this study, where in a small sample of cases, we looked to see if we could use Fourier transform patterns from imaging data to distinguish patients with fusions versus those that don't have fusions. And here you can see it much more readily, where the patient in red is the one with the fusion, and the other ones don't have the fusion. There are a large number of pharmacological agents now. We are into the third and maybe soon the fourth generation of FGFR and FGFR2 specific inhibitors in this disease. Infigratinib, shown here, was the first to be approved, the second to be approved. Pemigatinib was the first to be approved. These would be considered first generation. Futibatinib, which is a covalent inhibitor, was the third to be approved. and a number of other ones coming down the line. So in a very short amount of time, you can see that things are being used readily in clinical practice. Here's the study with infigratinib in patients with FGFR2 fusions. You can see from the waterfall plot that 60 to 70% of patients have some clinical benefit. When you use the RECIST threshold, we're down to 30% range that have a quote unquote response. These responses are reasonably durable. They last 6 to 7 months on a median basis. But unfortunately, response followed by progression is the name of the game. Toxicities are related to targeting the other FGFRs, so hyperphosphatemia, diarrhea. These tend to be off-target effects because these are not FGFR2 related. Things like ocular toxicity and skin and nail toxicity are very much on-target effects because they're related to FGFR2. A study had been started to do confirmatory work with infigratinib, but unfortunately this drug is now off the market for commercial reasons, so I believe it's been suspended at this time. Pemigatinib is the first inhibitor to be approved, although it was one of the later inhibitors to be studied, and there were 3 cohorts, one with fusions, one with other alterations, and one without FGFR alterations in the initial study. You can see from the waterfall plot, again, 40% or so response rate. 70 to 80% have some tumor shrinkage. And same kind of adverse events with phosphate changes, skin, nail, ocular toxicities, and some gastrointestinal effects. Ophthalmologists and dermatologists need to be part of the team managing these patients because of the on-target toxicities I mentioned. Futibatinib is a covalent inhibitor, so perhaps there could be some advantages where the drug binds and stays bound for a while, although for a daily administered drug, I don't think that's that critical. You can see from the preclinical data that it can overcome the vast majority of resistant mutation effects, although there are some that are able to escape, and these are the mechanisms of resistance even to the second generation inhibitor. Even in this initial study, there were a few patients that were treated who had previously received FGFR inhibitors that got futibatinib and had response, which validate the preclinical data I showed you. This has obviously now been approved, and the pivotal study was published in New England Journal and showed a higher than 40% response rate and reasonable durability that exceeds modestly the previous durability of responses that I described. So perhaps this might be, out of the 3 agents, the one people might preferentially use in the clinic currently. Mechanisms of resistance have been described, which are both on target and on pathway rather, and off pathway. On pathway pertains to polyclonal FGFR2 mutations where drugs are unable to bind for certain resistant clones. And obviously the off-target, off-pathway ones would be the harder ones to manage because they could be a heterogeneous group related to downstream events like PIK3CA or parallel events like HER2 or MET. There are 3rd generation inhibitors now, as I specified. So lirafugratinib, also known as RLY-4008. This just illustrates how effective these drugs are. Over 85% response rate in a small cohort of patients, but we're going from 30 all the way to 80 to 90. So clearly, just in a short amount of time, the amount of progress is accelerating. Another facet with identifying drivers, like I said, you could have allele-specific expression. And here's an example from, again, that Mayo TGen study where you have a mutation in ERRFI1 which is a negative regulator of many growth factor pathways such as MET and EGFR. And while the number of DNA events was few, at the transcript level, there were a lot of events shown in green. So this is also known as MIG6. And as I said, this is a negative regulator of pathways such as MET and EGFR. This has been described in some more basic studies in terms of what MIG6 does. And there have also been descriptions of mice that don't have MIG6 forming biliary adenocarcinoma. So it was surprising to actually see this in a patient. So there were no prior evidence to guide our decisions. So as I said, we know what the pathway does. So we went ahead and gave the patient erlotinib, and you can see that there was significant decrease in hypermetabolism and shrinkage of the tumor in just 3 months. The patient maintained this for about 6 months, but unfortunately then developed resistance and progressed, but at least the initial foray was successful. We talked about FGFR2 fusions and rearrangements, but you can have other ways in which the target can be affected. So here's a patient with cholangiocarcinoma, had profiling done, had received first-line gemcitabine-based therapy, and then progressed, and then the patient had an FGFR2 F276C mutation identified at that time. It was not classified at that time. It was listed as a VUS, but one could see that based on the literature, this was occurring in a place where there could be increased dimerization and enhanced activity. So we went ahead and gave this patient infigratinib, which was available through a study. And you can see here that the patient had significant reduction in their tumor burden, and this lasted actually for almost a year. And then we went back to the lab and demonstrated that F276C mutations lead to hyperactivation of the pathway compared to wild type. Now we know that there are about 2 to 5% of patients that have these gain of function mutations, and many of these are sensitive to FGFR inhibitors. So now you've increased your pool by 50%, going from 10 to 15% if you are able to integrate all of the mutations correctly into your pool. It would be nice if we could predict this already through other means, which include protein modeling and machine learning based workflows without having to do any functional studies. Now, when you look at cholangiocarcinoma and gallbladder cancer collectively, you could refer to them as biliary cancers. Even with just the panels that I described, which have 400 to 500 genes that are covered, you see that more than 50% of these patients have a target. So it's the most target-rich GI cancer, and you could think of it as like the equivalent of lung cancer in the GI cancer world from a precision medicine standpoint. What are some of these other events? IDH events are quite common, 10 to 15%. We know that this is a metabolic and epigenomic protein and enzyme. There already has been a confirmatory study with ivosidenib, comparing it to placebo. Here, patient advocacy played a big role. I saw Melinda is on the Zoom, so her team really had a big say in how the study was done, where even though placebo was used because crossover was allowed, this enrolled very rapidly and was fair to patients participating. There was a difference in progression-free survival, although it was modest. One can see that there's a group that's kind of like that tail akin to patients we observe on immunotherapy. So maybe 10% or so of patients have more profound durable benefit with these agents, despite the short PFS. Because a technique called rank preserving structural failure time was used, overall survival benefit could not be demonstrated due to the crossover. But had crossover not been allowed, likely you would have seen formal survival benefit that was statistically significant. These drugs have modest toxicities, mostly some myelosuppression, GI effects, and fatigue. Mechanisms of resistance are not well known at all. Maybe IDH2 mutations as one mechanism has been described. HER2 is another target, about 5% in cholangiocarcinoma. As amplifications, 2% as mutations, so 7% collectively. A bit higher in gallbladder cancer, about 15 to 20%. We see that the types of mutations in the HER pathway can vary. And there have been studies done both in the mutation arena and the amplification context. This is the Summit study that's now been published that Jim Harding led, where we look at activating mutations being treated, and you can see that they occur across the length of the protein, primarily in the kinase and dimerization domains. And the response rate is not that high. Maybe only half of the patients on the waterfall plot get some benefit, and even these can be short-lived. So here you can see that the patient got a response by 12 weeks, and by 24 weeks, they've already progressed. So durability is the big challenge. An agent called zanidatamab has also been described now with patients who primarily have amplifications being treated here. And this is an antibody that's a bispecific with one end that binds to the HER2, like trastuzumab does, and another like pertuzumab to the domain that dimerizes EGFR and HER2 on the same molecule. You can see that this has 60 to 70% of patients having some tumor shrinkage. The formal response rate was around 40% by RECIST. BRAF mutations, about 5%. This has now been approved due to the conduct of a basket study involving lots of different tumor types. So dabrafenib and trametinib were the agents used here. You can see 70 to 80% of patients have some clinical benefit. About 50% of patients have some RECIST response. And there are other targets being pursued with these type of basket studies, as shown here. And there are other designs that have been proposed where you even look at some level of randomization after you do the profiling in the second line setting. Although personally, I am not a big fan of these types of randomization experiments when we already have a pretty good idea that these drugs are active in a rare and lethal disease. A few other alterations shown here. As I described, there's a long tail of alterations with this disease. Liquid biopsies. I think it's already a tool of the present. So maybe the talks as they go along, I'll have to not make it sound so futuristic anymore. Big advantage of these types of assays, where you can either look at mutations or methylation markers or other aspects with just the analysis of a plasma sample, is that they're faster. You can get the results back in a week or less. They're much less invasive than getting a biopsy. You can analyze often without risk. Because they look at a sample that represents really the whole tumor in a way, they are maybe a better representation of tumor heterogeneity than a single biopsy. And you can use it to not only identify targets, but actually to assess response. I think scans will become secondary. I doubt we'll hear the term RECIST anymore in a number of years. It will probably be confined to the archives of medicine very soon. Like others, we had done some work trying to characterize biliary tract cancers with these modalities. And you can see that the most common alterations are things like KRAS and P53, FGFR2, IDH, the ones I described, and then a long tail. In patients where you have stable disease, so here's a patient with IDH1 inhibitor that I had on treatment, and you can see that they had stable disease after 7 months, but I couldn't really tell if it was their disease or effect of the treatment, but liquid biopsy really gave a clear picture. You see that all their alterations were not detected at that time, and this would give high confidence that they were actually responding instead of just having stable disease. So what can we use this for? Target detection, as I said, using these panels, hopefully can go even broader as time goes along. I hope to see less imaging because I think this is much more quantitative than RECIST, less prone to user variability and other facets, and it's faster and cheaper than getting a scan. We could use it as a discovery tool, although these are small bits of DNA in the circulation, so it's hard to be as comprehensive as you would with tissue. You could use it for minimal residual disease detection, and assays that are designed for early detection will be particularly important in patients that have known risk factors where you have higher incidence. Okay, so I said the last thing I'll talk about is the battle royale, which is going to be the big fight between N=1 medicine and evidence-based medicine. I imagine in oncology at some point, evidence-based medicine will become extinct once we have the appropriate tools that can truly be highly predictive for individual patients. So what do I mean by that? Well, there is a big foray into looking at functional metrics of response. So could you get organoids from a patient, or could you derive a xenograft? These take too long, or might be not high in take rate. So while these tools have been available, they haven't been used widely clinically, at least. But hopefully newer platforms like this will allow you to have good concordance between response you see ex vivo and response you see in vivo in the patient. So here you can see that even using these organoid models, you can predict if a response would have occurred. Here's some organoids that collaborators from our group had generated. This is one that had an FGFR2 fusion, and here we didn't use an FGFR specific inhibitor. We wanted to see if a drug like lenvatinib would work, and we saw that because it has some FGFR inhibitory effect, it was effective in these organoids. So again, these become tools for individual patients potentially. So given that this ex vivo facet is so critical, we set up a system called the ex vivo platform at our genomic center called the Mayo Clinic Center for Individualized Medicine. And what we have been doing in this effort has been integrated whole genome sequencing. We replace exome now with whole genome, mate pair sequencing, RNA sequencing, tools to integrate all the data akin to the ones I showed you earlier, and then a parallel sample where you cryopreserve it. It's not organoids, but short-term 3D cultures where you preserve the tumor microenvironment using hanging droplet methodologies. And then based on the sequencing data, you narrow down to a group of drugs that you want to test in the functional assay, and then the ones hopefully that you see there are the ones you would eventually take to patients in a CLIA setting as things progress. So this is no simple feat because it requires coordination amongst many different technologies and groups. Pathology, you need a lot of upfront specimen optimization because you're working off of small biopsies, attempting to do whole genome sequencing. Anyone who's done that would know how difficult this can be if you haven't optimized your sample. You can have molecular biology techniques, including things like flow sorting of nuclei to enrich, against giving you the ability to do more off of small amounts of material. Because you get so much data, you're not dealing with a report anymore. You're going to be dealing with the whole genome, and that requires not a tumor board, but what we like to call a think tank, where primarily the representation is from people with genomics experience, pathway experience, molecular biology experience, pharmacology experience, not just various specialties in terms of clinical medicine. Okay, so there's obviously a lot of moving parts, and one can see that this is a very complex, expensive endeavor, but what you get out of it is extremely high resolution. So just laying out the process here again: multi-omics, think tank, microcancers, make decisions here, test them out in these models, and then maybe take them back to patients in some instances. So here's a dashboard of the process to make sure it goes from acquisition of the sample all the way through delineation of recommendations. And this is something that bridges discovery science and the practice, and I'm showing only a small portion of the group here. It obviously requires a village. So all elements are critical. You can't discount even simple things that you might think like tissue processing in this paradigm. And like I said, you can test the drugs, and you have to use short-term ATP-based readout assays currently, although you can go into other ways of looking at effect other than cell death as you go along. We do see that these microcancers recapitulate the original tumor in terms of their genomic features on a whole genome scale. And just in the interest of time, an illustrative example, here's a patient who had some panel sequencing done, some prior therapies, received a drug on a clinical trial, which was not effective. So we have the full genomic map here in terms of all types of alterations, rearrangements, mutations, and copy number changes. We highlight some of the key ones, including some events that weren't described on the panel report that I showed you. And the functional testing allowed us to also use combinations of drugs where you don't have a biomarker perhaps that you might want to consider for use in such patients. So you can see that we've actually found that gemcitabine remained a drug that we could use here, and the drug that the patient went on actually was not effective. So this corroborated the findings from the functional assay, showing how important both functional and genomic data analysis is in making final determinations. I'll skip through this one here. It's just another case. So conclusions, future directions. Genomic profiling is the standard of care. So if you're not doing it, I'd love to hear your excuses, but there shouldn't be too many. And platforms like the one described remain discovery platforms. No matter how many cases you've done, you're always going to continue finding new things, because the tumor is not a static environment, but an evolutionary body. We will want to gain deeper understanding of things that are related, but outside of the realm of precision medicine, such as immunotherapy, where there will be that interconnection. And that will be definitely a big area. It's been hard to find biomarkers for immunotherapy response, of course. Liquid biopsy, I think you could already say that it is the tool of choice when feasible, and I hope to not hear about RECIST in the years to come. And functional models will become more and more critical, and I hope they will help displace evidence-based medicine in favor of N=1 medicine. So I want to first and foremost, thank patients and their families for their tremendous courage and generosity for making all the work I described possible. And of course, many funding agencies, including your own institution, for providing some support for some of the things I showed. With that, I'd like to close and thank you for your attention.
Thank you, Mitesh. That was a very great talk. Sorry, my camera is not working right now. So that was a very interesting forward-looking talk. And I have a specific question, which I thought was fascinating. Very early on, you talked about AI and prediction of FGFR fusion. And at the same time, we know that obviously one of the problems always is the requirement of tissue and how much tissue you have. And I was wondering, that study that you did, how does it compare to actually sequencing? So in other words, can you use less tissue potentially for AI than what you would need for sequencing?
Yeah, what I showed there was actually off of CT images. So actually, you don't even need tissue. You could.
Oh, sorry, that was off CT. Okay, I'm sorry. I thought it was of H&E sections that you would do.
Yeah,
AI.
no, it was just radiological images.
So, okay, so now I'm really going to challenge you now, because you just said RECIST will no longer be needed. But maybe you actually need the images to predict your genetic signature over time. And
yeah,
how that may change, then then you have to actually compare it to the liquid biopsy. And you may actually get, I would then argue, well, you just do an image and you can potentially get the signature plus the image in one test.
True. I think that's a good argument in favor of keeping scanning. Modalities don't tend to go away, right?
Right.
My feeling is it will just become less frequent. So do you really need to do it, every 2 months?
Yes.
Right. I doubt that, right? Once you know you're on the right path, maybe you can
of course.
check blood, right? Which we can't do right now, unfortunately, right? It would be outside of standard of care if you did that. Plus, it's not quantitative, so you could be a responder, and then if somebody measured it a little bit outside, you're a non-responder, right? So this definition from RECIST was always very arbitrary. So I think ctDNA will provide that quantification that's unbiased, that doesn't matter who does it, you're going to get the same answer.
Okay, are there any other questions?
Let me follow up with on another challenging question. So you probably read that recent paper about the NCI match trial, based on very similar strategy that you have done using genomic sequencing of the tumor and identify driver mutation and then enrich those for targeted drug. And now we have the data, at least the 27 of those trials was enriched for the drug, not working, nothing works. So enrichment doesn't necessarily improve. So I think one of the interpretations from those negative results is possible. One is the functional driver, a definition of a driver, whether those are in the clinically, as functional, or supposed to be laboratory functional. And clinically it may not be functional, right? Part of the reason is that because, as you see from your genome sequencing data, that some of the, not all cells carry that mutant allele. So tumor cells that do not carry those allele will escape, will expand. And then you say that the FGFR2 inhibitors actually, most of the people that will relapse after 6 months. You have initial response and then relapse. What's your thoughts in the future? How do you move forward on that?
Yeah, so that again goes to the last part I was trying to highlight, which, and again, I didn't show enough data to really make an extremely strong case, but every patient's tumor, because it's highly distinct, if you use the bucket and basket approach, right, where you say FGFR2, you're only treating patients for one of the things they may have in many instances, right? They may have a second event. So if a patient has an FGFR2 fusion and a PIK3CA mutation, they should probably get a combination. But guess what? In that trial, they're only going to get one drug, knowing that they're already going to fail likely, right? Combo match is an evolution to that, right? But how many combinations can you come up with? There's not a way to have enough arms unless those events always go together, right? So the reality is at some point you will have a toolkit of things that are pharmacological, inhibitors or activators of various pathways, and you'll just have to choose from them, and you might not know the toxicity, so you'll have to tweak things in individual patients rapidly to arrive at a combination that's tolerable and likely going to be effective, because you're going to have to hit all the key events, not just one or two.
So how do you evaluate those? The combination obviously is one way to go forward, but in terms of combination, you have to evaluate it for each combination. How do you measure the objective response for each individual patient if you don't necessarily design in some sort of randomized?
Yeah, so that will require us getting rid of this idea we have about what is design of trials, right? Oh, we need certain number of patients, we need certain amount of response above the hypothesis. I don't think we'll need that. We'll use these functional tests hopefully to see, are our odds going to be high if you use one or two? Should you even use the combination? Or are you going to do okay with one, right? If you need to, then you're just going to have to go into that patient with that combination. And there could be some challenges with the dose, right? What dose do you use? Well, you pick some arbitrary doses and then change them over some period of time and get to a dose they can tolerate. Having confidence that your functional test, if it's reliable enough, is going to translate into benefit for that patient. So there will be no trials of 50 patients anymore. It's N equals 1 for every patient.
So you mentioned about the deep sequencing of the genome, carry out a more sequencing and whole genome sequence, deep sequencing. Are you able to actually use deep sequencing of the tumor to find potential founder driver that could possibly be as earliest events that you could stop them?
Yeah, no, absolutely. I mean, we of course know many of the genes from the cancer gene census, but in that study that's still ongoing that I described, in the 50 or 60 patients that we've evaluated thus far, I still keep finding new things that have not been described in cholangiocarcinoma as potential drivers, right? So that's why these type of platforms remain ongoing discovery efforts.
Okay, thank you.
Okay, are there any other questions? Okay, if not, Mitesh, thank you again. This was a very, very provocative, forward-looking talk, which I enjoyed a lot. Thanks so much, and we will see everybody soon on this channel. Bye-bye.
Okay, thank you for having me.
Bye-bye.
Take care. Bye-bye.